So hello everybody to this nanostring webinar on innate immune activation in Alzheimer's disease. My name is Michael Heneka and I'm working at the German Center for Neurodegenerative Disease in Bonn and as well at the Department of Neurodegenerative Disease and Geriatric Psychiatry of the University of Bonn. So why Alzheimer's? We currently have about 35 million dementia cases worldwide and expect a rise to 150 million in 2050. In Bonn, in our department, we see about 800 new cases per year, and below you see analysis of the past decade of the World Health Organization showing that we were quite capable of dropping mortality due to various diseases, including prostate cancer, heart disease, stroke, HIV, but at the same time we have a dramatic rise in death from Alzheimer's disease. And that was the reason that actually the WHO made dementia and Alzheimer's #1 public healthcare issue in 2012. So we have currently no acute or preventive therapy available, and I believe that our understanding of the pathogenesis, the genetics, and the risk factors are all critical for the development of preventive, interventional, or acute therapies for Alzheimer's disease patients. So this is a rough schematic what we believe is going on in this disease. There's a precursor protein called amyloid precursor protein, APP, which is cleaved by various secretases, and the sequential action of beta and gamma secretase are yielding amyloid beta monomers, of which we know best the 1 to 40 and 1 to 42 amino acid length peptides. And those should leave our brain to about 25% via the CSF or 25% via the glial lymphatic system, while when they accumulate, they are going to be removed by microglial cells through various mechanisms of degradation. If they further accumulate into oligomer structures, they form high molecular accumulations which ultimately deposit in so-called A-beta plaques. These A-beta plaques are surrounded by astrocytes and activated microglia cells as part of an innate immune reaction, and ultimately the neurons show tau phosphorylation and so-called neurofibrillary tangles, which are showing dying or at least dysfunctional neurons. So what about innate immunity? We are constantly in our environment surrounded by viruses, fungi, and bacteria. And what the upcoming little video will show you is a bacteria which is going to hunt for an innate immune cell, which is going to hunt for bacteria. And you will see that this macrophage is following the bacteria throughout the cell culture, making a lot of clever decisions guided by surface receptors in order to finally hunt down the bacteria and ingest it. So this innate immune system is our first line of a defense against viruses, fungi, and bacteria. Play video. So these innate immune cells do not only exist in the peripheral bloodstream, they also exist in parenchymal organs, including the brain, where we call them microglia, alveolar macrophages in the lung, copper cells in the liver, mesangium cells in the kidney, Penney cells in the gut, and bone marrow macrophages, of course, in the bones. Now, one of their principal action is to protect these organs from infection, but they also contribute to organ structure and function. This is a frontal cortical section, which is stained for Congo red and MHC class II positive microglia cells. And actually, Congo red has been long before it was developed or used as a stain for human brains, developed for something completely different. Paul Bötticher in 1870 actually developed this stain to color shirts. And because the region in Germany which that has been developed is very rainy, it turned out that this would not stay and being washed out by rain. So they put it back onto a shelf until by accident, it was found that it also stains bacteria. But he went to Berlin and licensed Congo red as a bacterial stain. Now, what is the commonality between a human brain and a bacterial surface? This came out by work from Matthew Chapman, published in Science in 2002, where he described with his group that various bacteria, including E. coli, Salmonella typhimurium, or Staph aureus, which are all very common to us, carry beta sheet-structured amyloids on the surface. So these beta sheet-structured amyloids are very similar to the beta sheet-structured amyloids which are depositing in the brain of 80 patients, which can be seen here. Now, this could mean, as a hypothesis, that this myeloid cell here has been taught by evolution over millions of years that a beta sheet structure is nothing else but a pathogen, and for that reason it has been equipped by nature with various surface receptors, collectively denoted as pattern recognition receptors, including TLRs, CD47, CD36, and scavenger receptors. And if those receptors are ligated, there are two principal actions. One is the induction of innate immune signaling pathways, which are leading to the production and release of complement factors, chemokines, and cytokines. And another reaction, which is a phagocytic uptake reaction, in order to clear the debris which has been just generated. So if microglia cells get activated, these usually high arborized cells are retracting their processes, which are there to scan the immediate environment. And you can see that the plaque-residing microglia have lost most of these processes. Now, next to the inflammatory reaction, which I just described, the microglia cells also stop to do a lot of sensible things, including producing BDNF and other neurotrophic factors. They stop reducing, or they stop synaptic scaling, and they're also less efficient in clearing the debris from the brain. Now, one early indication that this microglial inflammatory reaction could be of significance to Alzheimer's patients comes from pathological work by Joseph Rogers and others, where they showed that actually the difference between 80 cases with a history of dementia and patients which have a high amyloid load in their brain, which are described as HPCs here, is the number of activated microglia cells at the plaque. In other words, when 80 cases were compared with high plaque normal cases without any history of dementia, the major difference was less microglial activation in the high plaque normal cases. Now, next to amyloid beta, the brains of Alzheimer's patients are bearing a number of other and sometimes unexpected danger-associated molecular patterns, and those include, for example, chromogranin A, which has been shown to be as effective in inducing, for example, nitric oxide as a sign of innate immune reaction when compared to lipopolysaccharide. So there is no principal difference between an LPS stimulus or a chromogranin A stimulus. There are, for example, MRP14 as another signal in the brain, and here you can see that MRP14 negative mice on an APP-PIS1 background produce by far less TNF-alpha and interferon gamma at the same time. Those microglia cells are more effectively clearing beta amyloid in a cell culture experiment. And this type of inflammation in humans can be visualized by PK11195 PET, which you can see in this 80 case versus an age-matched non-demented control, where there's only vascular signal visible. Now, what is the principal effect of inflammation on the brain? First of all, there's a functional effect, and we know that cytokines, including L1 beta, interferon gamma, TNF alpha, or nitric oxide, can suppress hippocampal LTP. And hippocampal LTP is not the equivalent to memory formation, but it's still an important mechanism for information storage and consolidation. There are a lot of mechanisms, of which I just want to point out three. There is this synergism with excitotoxicity, which has been well documented by work from Sandy Hewitt, where in an experiment, NMDA or an inflammatory stimulus alone was not great in inducing LDH release and neuronal cell death. The combination of both were really effective. Now, there is a more recent paper showing that microglial cells can engulf synapses when they are decored with complement 3 in a CR3-dependent mechanism. And Jonas Neher and Guy Brown have described a mechanism by which microglia can take up living neurons if they don't present so-called "don't not eat me" signals on their surface. Now, what is the time course of this phenomenon? When we believe from various papers and PET evidence that beta amyloid deposition could start as early as 2 to 3 decades before the occurrence of first clinical symptoms, then this neurodegenerative mechanism is actually way before the patient would come to clinic reporting short-term memory deficits in a phase which we call mild cognitive impairment or in full AD. But nevertheless, knowing that beta amyloid deposition is such a strong danger-associated molecular pattern, that would mean that all the other mechanisms thereafter are taking place in the presence of inflammation. Now, this comes on the background of various SNP polymorphisms, which are coding for genes that are important for either synaptic function or innate immune signaling, and of the latter, TREM2, CD33, BIN1, and others are prominent examples, but also ApoE4 has an impact on microglia function, and even presenilins may take part with some of their substrates to microglial phagocytic clearance of beta amyloid. Now, next to this genetic background, which certainly will influence the degree of innate immune activity and capability, our lifestyle factors may do the same. And here you can see that immunosenescence during aging is certainly a strong risk factor directly related to a dysfunctional immune activation. We know that systemic infections can be a trigger or a motor for cognitive decline. Midlife obesity in young men between 40 to 45 years is a risk factor that increases the risk for Alzheimer's disease up to fourfold 30 years later. And fat mass is just a constant source for cytokines, as a certain percentage of this mass is activated macrophages. Periodontitis seems to be a contributing factor. Sedentary lifestyle is an immune compromise, and brain trauma is probably the most direct induction of brain inflammation. All these factors here share increased levels of the release of immune factors and immune activation, and may be therefore a risk factor. Now, Midlife obesity is one of the named factors. This is an example where you can titrate your risk to develop Alzheimer's disease 30 years later by your sagittal abdominal diameter and BMI, and you can see this goes up to fourfold. The conclusion of that work is that midlife obesity increases the risk to develop dementia and AD independently from type 2 diabetes or any cardiovascular disease. This work by Jack Ivashina, published in JAMA in 2010, shows that people who had a moderate to severe septicemic event would have a higher likelihood to present with cognitive impairment thereafter and probably develop dementia and after this inflammatory event. And it's in fact something which we see in the daily clinical routine very often, that people would present after a strong pneumonia or another type of systemic inflammatory event with cognitive decline and accelerated neurodegeneration. Now, there were a lot of clinical studies or epidemiological observations which would indicate that, for example, the intake of non-steroidal anti-inflammatory drugs would be protective or at least reduce the risk and delay the onset for Alzheimer's disease. One of these studies was published by Innate and colleagues in the New England Journal of Medicine in 2001 and showed that the long-term medication with diclofenac, ibuprofen, indomethacin, and naproxen would effectively reduce the individual risk to develop Alzheimer's disease. There's a lot of trials which have been brought evidence together which points into a similar direction, but nevertheless, none of the acute treatment studies with AD patients have brought any positive result so far. So there's a problem, and maybe we have to rethink why the anti-inflammatory treatment therapies which we have tested so far were not effective. So what could be the problems? First of all, we have to think, how was this inflammatory activation coming along in the brain? Is this a linear development? Is this an asymptotic development or an exponential development? And I suggest that this none of any of those. I believe inflammation may run in waves, like in a normal skin wound where inflammation is defending our body from bacterial invasion. There's an increase, there's swelling, there is redness, there is pain, and at a certain stage, resolution. So probably what we see in the AD brain, that we have an early inflammatory rise being caused by A-beta oligomers, fibrils, aberrant RNA or misfolded proteins and peptides, which initially may compromise clearance, start to induce proliferation of microglia, and decrease trophic factor supply. Then we have the release of further danger-associated molecular patterns, such as chromogranin A from neurons, MRP14, heat-shock proteins, which may contribute to the aforementioned mechanisms, but also lead to neuronal dysfunction and damage. And ultimately, the loss of neurons is going to change the neurotransmitter levels, and we know that various neurotransmitters are actually guiding or regulating microglial activation, proliferation, and senescence, and so fundamental changes of neurotransmitter profiles may impact on another kind of inflammatory wave under those degenerative circumstances. Now, it's important to note that the time point of investigation will influence the nature, quantity, and quality of inflammatory signals detected, and thereby also lead to a biased identification of possible therapeutic targets. One could probably hit a target at its peak or at the decline, or in the mid of it, and the interpretation will be very different. So all the longitudinal data we are still missing need to be generated, and we need to be very careful with cross-sectional analysis, which we very much rely on to date. Another confound with microglial activation is that we have quite different microglia depending which brain region we are looking at. These are paper by Gravett and colleagues from 2016, where transcriptomal differences were shown between microglia isolated from the cerebellum, the striatum, the cortex, or the hippocampus. So what could be the basis of this difference? And again, very interesting review article by Jennifer Pocock and Helmut Kettenmann, published in TINS a time ago, shows how many neurotransmitter receptors are actually carried by microglia cells. And this is an example how, for example, noradrenaline through adrenoceptor ligation can regulate microglia reaction towards amyloid beta. We see a ribonuclease protection assay and under control, a beta stimulation or co-stimulation with norepinephrine conditions, and it becomes quite clear that norepinephrine suppresses the activation of these microglia cells by A beta 1 to 42. It does not only suppress the inflammation, it also increases the interest and the microglial clearance capacity in the whole brain of mice. This is an experiment shown by two-photon laser scanning microscopy, where microglia cells surround a little A beta deposit in norepinephrine intact conditions, while under DSP4, which depletes norepinephrine from the brain, you can see that at a very tiny plaque site, microglia cells assemble at time point 0, 6 weeks after this plaque has grown substantially, and there's no microglia cells which would try to limit this or take amyloid beta down. This actually can be rescued by treating these animals with L3-ODOPS, which is a direct noradrenaline precursor, being converted by dopa-decarboxylase and able to rise cortical and limbic system levels of norepinephrine after use before treatment. Now, next to these probably changed levels of microglia guidance and regulation through neurotransmitters, microglia cells undergo cell death and proliferation in APP-PS1 mouse models and AD patient brain, as indicated here. So on the right side, this schematic shows an increase in cortical and hippocampal microglial proliferation over time, and this can be found in the brains of patients back. So interestingly, we have always believed that a microglia cell which would encounter beta amyloid peptide would probably show an acute inflammatory reaction and turn into a chronic active microglia cell. The question is if this is a correct model, and I want to suggest that we have to think about microglia population change by accelerated proliferation. So if microglia cells which are populating our brain very early and form this very nice lattice encounter an early amyloid beta deposit, some of them will undergo death, some of them will undergo proliferation and activation surrounding this amyloid beta deposit, and as time persists, many different generations of microglia cells may actually arise. And this would mean that we are not going with one microglia population, but with more. This is one of many data sets showing IBA1 BRDU incorporating cells at a methoxy XO4 positive amyloid beta plaque. Now, over time, when we start with surveying and clearing microglia cells over time under neurodegenerative conditions, we could have multiple generations of microglia cells, which would then become harder to target because the cells which proliferate into such an inflammatory environment probably acquire functions and actions way different from those which have originally populated the brain. Now, there are a lot of studies on murine microglia, so we wanted in our group to focus on functional signaling pathways which are really important to human microglia. And this paper by Bart Eggen and Eric Bodeke has nicely combined for different data sets to generate what they call a human microglia core signature. And what you see right in the center of this is, what you see in the center of this are the knot-like receptors and the inflammasome, as well as their products IL-1 beta and IL-18. Now, the NALP inflammasome in the brains of mice and 80 patients is restricted to microglia cells. It's highly controlled. It requires a step of transcriptional activation, ligation of a toll-like receptor or a cytokine receptor, which then induces the generation of transcription factors and cytokines and chemokines, also the auto-inhibited NALP3. And this is followed by next step of post-transcriptional activation, various steps that license the activation of the NALP inflammasome and formation of an ASC-SPEC, which then has the active caspase 1, which is able to convert proforms of IL-1 beta, IL-18, and gasdermin to either the active gasdermin P10 that forms the pore or the active cytokines, including IL-18 and IL-1 beta. First thing we did was we analyzed whether this mechanism was actually found in human AD, and here you can see a microglia cell containing an ASC-SPEC in green. In a more quantitative way, we assess the activation of the inflammasome by immunoblotting for pro and cleaved caspase 1. And if we do so in brain samples from age-matched non-demented controls in 80 cases, you'll see that there is a strong increase in cleaved caspase 1 signal in 80 patient brains. And this phenomenon is not restricted to 80 cases, can also be found in patients which suffer from MCI, so the pre-stage of Alzheimer's disease. Now, importantly, the pathway of the NALP inflammasome activation is highly conserved between mouse and man, and Picard, also called ASC, IL-1 beta, caspase 1, and NALP are expressed mostly by microglia and macrophages in both species. Now, we used the APP-PS1 transgenic mouse model because this is, in our view, a murine model that is ideal to investigate the consequences of innate immune activation. You can see that these green amyloid deposits here are massively surrounded by red microglia cells in this model. Using this APP-PS1 mouse model, we then crossed NALP knockout animals into it. And one of the important findings was that while the APP-PS1 animals were characterized by reduced trophic factor secretion, NOS2 expression, and IL-1 secretion of microglia cells, in this case on an mRNA level here, we found that this was all converted by the presence of NALP knockout animal background. So instead, microglia from APP-PS1 NALP knockout mice showed increased levels of arginase 1, FIS1, and IL-4. And most importantly, they also showed a strong increase in IDE. And IDE is one of the key enzymes of extracellular amyloid beta degradation. Now, when we looked and under high magnification to amyloid beta deposits, we found that in APP-PS1 transgenic mice, the microglia cells would surround the plaques but would not very much interact with them and seem to be paralyzed, while in the case of the APP-PS1 NALP deficient animals, the microglia cells were really taking the amyloid beta apart and sending lassos into the plaque, which were filled with beta amyloid, indicating active phagocytic clearance. So, in order to assess this possible clearance mechanism, we developed an assay which allowed us to quantify microglial phagocytic uptake of beta amyloid, and we used methoxyxo4 injection, waited for 3 hours, explanted the brain, isolated the microglia, and faxed them for the methoxyxo4. Now, what we found was that in contrast to APP-PS1 knockout mice showed a much higher amount of methoxyxo4 containing CD11b positive microglia cells. And there was over a number of 8 animals, twofold increase in phagocytic clearance. Cells were then isolated and analyzed, and we found that we can overlay methoxyxo4 with another amyloid beta specific antibody, IC16, within the CD11b positive immune cells, showing that over the time of 3 hours, these cells in fact had taken that amount of amyloid beta. This increased clearance in response to NALP deficiency reduced the hippocampal, frontocortical, and motor cortical amyloid beta by about 70 to 80%, and this result was also confirmed by ELISA measurements. So in order to assess a spatial navigation memory, we are using the so-called Morris-Water maze test, where animals follow cues in a water bath seeking a hidden platform, as you can see in this little movie. And after a while, the mouse will find the platform. The parameters we test are actually the time to platform, which is called latency, as well as the distance the animal are swimming before they actually sit on the platform, as you can see now. So this is being analyzed at 8 consecutive days, and in this experiment you see that wild-type and NALP deficient animals very quickly learn the task, and then they consolidate that information, while APP-PS1 animals in red don't learn much over these 8 days. APP-PS1 NALP knockout animals were almost as good, despite they were 18 months of age, and so they were almost performing as good as the wild-type or NALP knockout mice. On day 9, we perform a probe trial, so the platform is being removed, and you can see then whether the animals have spent time in the first quadrant, remembering that the platform had been there. And what you see on the right side is that wild-type and NALP deficient animals do so quite good. The APP-PS1 mouse has a more random search strategy at day 9, while the APP-PS1 NALP deficient animals actually very much restricting their time to quadrant 1. So the next interest we had were to analyze whether innate immunity would actually contribute to disease progression, because we all know that amyloid pathology spreads within the brain area and from one brain area to another, and the brain is not destructed in a day, a week, or a month, but it takes several years to take it down. And so the current model of spread of pathology implies that fragmentation of a fibril is then followed by nucleation and growth, and once a fibril has a certain length, it would fragment again and would represent a new nucleus. Following this, we asked whether innate immunity would contribute to disease progression and spreading of pathology. Now, we did so because in a collaboration with Fred van Leuven in Belgium, we very early observed that when we compare APP V717I, so-called London mutation, single transgenic mice, we don't observe any extracellular amyloid beta deposition at 3 months, but we already see focal microglia and focal astroglia activation. And interestingly, this microglia activation was characterized by Al1 beta positivity. So the microglia cells were here positive for a factor which we connected to the activation of the Inflammasome and ASC-SPEC formation. So microglia cells, in fact, when they get overactivated and exposed to pro-inflammatory cytokines, will undergo a so-called pyroptotic cell death and release the ASC-SPEC. And in fact, in AD patient brains, as well as in APP-PS1 transgenic mouse brains, we can see ASC-SPECs within microglia cells, but also in the extracellular space. So we learned to identify and track these ASC-SPECs in cell culture and actually induce microglial pyroptosis by LPS priming and exposure to nigericin or ATP. And then we were aiming at showing that beta amyloid would be a fantastic inducer of microglial pyroptosis, and we kind of failed here. What we found is that as soon as an ASC-SPEC would be released by a microglia cell, the beta amyloid peptides would be very quickly bind to this ASC-SPEC. And you will see in this little video that microglia cells which release an ASC-SPEC show the direct binding of the red labeled amyloid beta to the SPEC. Play video. We next looked at this on an EM level, and you can see here amyloid beta 1 to 42 alone, ASC alone, and hopefully you would agree that amyloid beta and ASC then are kind of very tightly bound into the co-incubation experiment. And that leads to the hypothesis that once a microglia cell would undergo activation, inflammasome formation, and ultimately microglial pyroptosis, the released ASC-SPEC could interact with the beta amyloid peptides and represent a matrix which would be the initial step in plaque deposition. And to assess this, we utilized the thioflavin T fluorescence assay. In green, you can see the aggregation curve of amyloid beta in the absence of any ASC, and then this shows the conversion from the monomer to the aggregate over time. And it's easy to see that adding increasing concentrations of ASC-SPECs to this causes a very strong shift to the left, indicating classical cross-seeding. Looking at the same or a similar experiment read out by immunoblot, you can see that after 24 hours, in the absence of any ASC, there is hardly oligomer and only few A-beta fibril formation, while under increasing concentration, there is strong formation of fibrils and A-beta oligomers. This is an experiment which used amyloid beta 1 to 40, and it looks very much the same for amyloid beta 1 to 42. Now, in another experiment, we used a co-sedimentation assay where we had 3 different conditions: amyloid beta 40 or 42 alone, and the presence of ASC or ASC alone over 6 hours. And what you see is that everything starts in the supernatant at time point 0, and after 6 hours, the ASC has dragged the entire amyloid beta peptides into almost artificial pellet or plaque structure. Now, real plaques show binding of IP'd ASC to amyloid beta in an age-dependent manner in mice at 8 and 12 months. When we look to the plaque histology, we found that ASC actually sits in the center of the beta amyloid plaque, and the 6010 positive amyloid beta is surrounding this core, a little like in the movie I've just shown you. So that would not yet indicate that the presence of the ASC speck is the start of the plaque deposit. And in order to test this hypothesis, we employed a model which has been nicely introduced by Matthias Jucker and Larry Walker and summarized in a Stellar Nature article in 2013. So the basic principle of this experiment is to compare solvent control and APP-PS1 lysate injection in the hippocampus of opposing brain hemispheres, and to wait a certain time period to read out the spread of A-beta pathology. And interestingly, a synthetic A-beta is almost not seeding in this model, suggesting that amyloid beta itself may be different, or there may be one or further endogenous cofactors which are responsible for this observation. Now, this experiment shows you the comparison between wild-type brain lysate to APP-PS1 lysate injection in an APP-PS1 mouse model, and we actually verify the results published by Matthias Jucker and Melanie Meyer-Luhmann in 2008. The open to the full bar shows a twofold increase in area and the number of A-beta plaques. In the analysis of fibrils and oligomers, you see a strong increase. Total A-beta is dramatically increased, no effect on APP processing or APP itself. Now, as a comparison, we looked at brain material from non-injected mice, and here you can see that wild-type mouse brain lysate injection is not having a great effect. So the non-injected animals principally the same as the wild-type lysate receiving mouse brain shows. And the next step was to do this experiment in the absence of ASC, and this took the entire induction or increase in area and A-beta deposit number away, but more importantly, it prevented the formation of oligomers and fibrils and did not increase total amyloid beta. So this experiment suggests that, at least in this model, the presence of ASC is required to induce the spreading of A-beta pathology. Now, what about humans? When we IP for ASC and we plot for amyloid beta, we found A-beta bound ASC only in the brains of 80 cases, but not in age-matched control. controls. More interestingly, when we splitted the fluffy fiber compartment from the core of the plaque and analyzed brains of MCI cases, we also already found a co-localization of ASC and A-beta in the core of MCI plaques, while the only difference to Alzheimer cases was that there was more A-beta in the fiber compartment. Now, looking at these plaques, most of them show an ASC-positive red core and 6010 green material surrounding it, again supporting the view that the early innate immune activation and the pathologic release of ASC specs could be one dimension which is important to plaque deposition. Now, is this current finding? I don't believe so, because in this picture from a frontal cortex of an AD case where we stained for ASC and I-beta, you see a lot of ghost microglia which have undergone pathologic death. You see released ASC specks all over. But the little inlet on the right side, which is taken from a publication by Alois Alzheimer and Franz Nüsse in 1911, shows you that you can see all these destructed microglia already in their publications. So I think they were quite aware of the microglia pathology which takes place in this disorder. Now, what about tau? One of the interesting questions along was whether tau next to A-beta would also be able to induce inflammasome activation. And in order to test this, we used brain lysates derived from tau-22 animals, which were provided kindly by Luc Bouet from Lille in northern France to us. And you can see that there's a minor increase in L1 beta release by microglia cells, which are exposed to tau-22 brains as compared to wild-type brains. So, encouraged by this experiment, we actually went on and used tau monomers, oligomers, and fibrils for inflammasome induction, and here you can see by readout of L1 beta production that monomers and oligomers were quite efficiently inducing the L1 beta release and less so fibrils. In all cases, however, this was dependent on the presence of the NALP3 inflammasome. This is further supported by the analysis of cleaved caspase-1 in this capillary Western blot, where you see that tau monomers and oligomers induce a strong caspase-1 cleavage, while fibrils are by far not as efficient in doing so. So the animal model actually was carrying mutations from patients, families suffering from frontotemporal dementia. So we set out to test whether patient brain samples from those patients would show an increase in caspase-1 cleavage, and they do so. FTD obviously is characterized by a very strong induction of the NALP inflammasome. It also shows strong increases in the P17 band of L1 beta, so that we are sure that this system is generating this key cytokine in the brains of FTD patients. Now, it's always good to know that your mouse model, in this case the tau-22 animals, mirrors key elements of the human disease, and you can see that neuronal pathology, especially neurofibrillary tangle formation, is displayed quite nicely in the animal model generated by Luc Bouet and his group. These animals also show caspase-1 cleavage, and induction depending on the presence of the NALP inflammasome. Using these mice and analyzing their brains with a nanostring neuroinflammation panel, we found that in our hands, the first on first sight, the PCA analysis would cluster for age and not so much for being a tau transgenic, which just on first sight was disappointing. But by looking more closer to these results, we actually found that what we detected was the induction of a senescence-associated secretory profile or its inducers along with various inflammatory mediators. Now, that encouraged us to cross the tau-22 mice in NALP or ASC knockout animals, and so we waited for 8 months to read this out by 88 pathology. And you can see that ASC knockouts and more so NAP knockouts showed protection. And quantifying a larger number of mice showed that this was for real, and in particular, NAP knockout would be protective for this type of pathological tau change in the tau-22 mice. Well, but because it's unlikely that one pathway is important for both the A-beta as well as the tau pathology, we repeated this experiment, waiting a little longer in the hope that the phenomenon would fade out, but it didn't do so. After 11 months, the protection was even stronger in the ASC and NALP knockout tau-22 mice as compared to tau-22 animals. So we had to come up with an explanation, and as a recapitulate, tau phosphorylation is guided by various phosphokinases opposed by one or more phosphatases. And if the activation of phosphokinases is stronger than the phosphatase activity, this leads to neurofibrillary tangle formation and neuronal destruction. So we tested this in the brains we had, and what we found was that we actually reduced the amount of inactive PP2A, while at the same time reducing active chem kinase to alpha and to a lesser extent GSK3 beta. Now, importantly, we were able to show that the modulation of chem kinase to alpha is critical for the observed protection because its target tau phosphorylation at serine 406 is strongly reduced in tau-22 mice when they were NALP or ASC deficient. So ultimately, we wanted to bring this on a cell culture level in order to delineate the involved pathway, and what you can see here is that microglia cells, which have been generated from wild-type NALP knockout or ASC knockout, can be stimulated by a certain protocol, LPS and ATP, to induce the NAP inflammasome. Then we take the conditioned media, transfer them on primary hippocampal neurons, and analyze those at 24 hours. So in this experiment, we found that inflammation strongly induces phosphochem kinase alpha, which is depending on the presence of ASC and NALP, while co-incubation of hippocampal neurons with the interleukin-1 receptor antagonist would strongly reduce this observation as the use of an IRAK4 inhibitor or a MEK1/2 inhibitor. So furthermore, we found that tau-5 and paired helical filaments are strongly induced by this inflammatory stimulus and reduced in the absence of ASC and NALP knockout. On a behavioral readout, these animals do not show a learning defect. So over the 8 days of learning, the animals are indistinguishable from wild type or ASC or NAP knockout. But at day 9, when we test with a probe trial the memory, we can see that while wild type ASC or NAP knockout animals spend way more time in the quadrant where the platform had been located, the tau-22 animals seem not to remember and have a random search strategy. While when crossed into ASC or NAP knockout, they regain this function, and the latter groups are almost as good as the animals which do not carry a tau-22 transgene. So the ultimate question was how to get from A-beta deposits to neurofibrillary tangles, and whether innate immune activation, in particular now inflammasome activity, would contribute to A-beta associated and secondary tau pathology. And to address this, we used a model introduced by Jürgen Götz when he was working in the lab with Roger Nitsch, and this paper has been published in Science in 2001. And what Jürgen did was he compared APP lysate injection with solvent control, and in tau-22 mice we did the same to test for spread of tau pathology, which in fact can be seen in the first 2 panels, wild type to APP-PS1 injection, so 88 staining readouts. This is the red against the red bar in wild type versus the red bar in APP-PS1 brain homogenate receiving hemisphere. However, when we do this in ASC or NAP knockout mice, this increase is completely gone, indicating that between the deposition of amyloid beta and the spread of tau pathology, inflammation could be an important contributing factor. So in summary, we come up with a model where microglia cells are undergoing inflammasome activation in response to pattern recognition ligation. They form a gasdermin D pore and release L1 beta and IL-18. These mediators, especially L1 beta, seem to bind to the L1 receptor, inducing a MyD88 MAC1-2 dependent chem kinase phosphorylation, which then phosphorylates the respective sites at the tau, leading to paired helical filament formation and neurofibrillary tangles. And with this, I would like to summarize. I hope I could convince you that beta sheet structured amyloids are a known danger signal to myeloid cells, including microglia. During waves of pro and anti-inflammatory changes, several other dams may arise in AD patient brain and maybe also in other degenerative diseases of the brain. The NAB inflammasome pathway is highly conserved between species and strongly increased in brains of AD patients. Experimentally, in murine AD models, NAB inhibition is protective. ASC specs are released by microglia pyroptosis, and they bind to A-beta peptides and increase their propensity to aggregate in a concentration and time-dependent manner in vitro and in vivo. And the NAB3 inflammasome knockout reduced tau pathology in tau-22 mice. This effect is likely to be mediated through the modulation of tau kinases and the phosphatase PP2A. And ultimately, I would like to thank all the people in my group and our great collaborators, and of course the people who have paid for this work. Thank you for your attention.